Perfect. So if you are going to share a presentation on the screen, I will recommend Prasad, what you can do is you can tag speakers and the presentation. You can pin these four windows so that everybody will uniformly see these four windows only, rather than switching between multiple
Yeah.
windows. And then of course, individually people will still prefer what they want to see locally there. But in our recording, you will always see only four people on the screen, one presentation and three speakers. So you can
Yes.
pin them on the screen.
You can see the presentation, right?
Yes, very well.
So I thought this is a good first slide.
Yeah.
Title.
So we are 3 minutes past the time we set for. So I think let us respect people who joined before time and on the time. I always really appreciate people who are spot on. So let's get started. I will set a context and we'll get moving. So I want to formally welcome all of you, all my participants, Manojji, Udayji, and Dr. Bunde. This is the second edition of this brand new GMG Select Talk relaunch. So we were doing this GMG Select Talk couple of years back, but that was more focused on different countries rather than on the topics. So when we spoke about this, the topic came out saying that shall we focus on the technology adoption by industry rather than just talking about the technology alone as a standalone topic. And that's how this concept was curated. We conducted our first select talk on supply chain, how AI is making supply chain smarter and how industry is consuming AI. So we thought that's going to be a very interesting for a lot of audience, that technology is finally a consumed for the betterment of an industry. And then the second topic came up that let's start talking about the health tech and let's start talking about the drug discovery, etc. And that's why all of you are here. So I formally welcome all my esteemed speakers. Manojji, thank you very much for taking effort and coordinating this. And you are going to be the navigator for the rest of the time and going to navigate through the challenges and the entire conversation. But it will, I have seen you doing this. You're seasoned at it. So I'm very confident. Thank you very much for doing so. Dr. Khire and Dr. Bonde, very warm welcome to this GMG Select Talk. You have been the stalwarts, you have been SMEs, decades of experience. So I see at least 100 year of together experience here on the screen as we commence this conversation. For those who have joined new to this conversation, Garje Marathi Global is a not-for-profit organization which works for entrepreneurship development, works for the professional next, and help students to grow in their careers. We are not-for-profit registered organization across continents. It is registered as not-for-profit in 7 nations, and we were able to connect about 20,000 like-minded professional people on this network. Little more about Garje Marathi. Garje Marathi started as a conversation from the people of Maharashtra going abroad and settling wherever they are. So Maharashtra is a common thread. Marathi as a language is definitely a connecting factor, but it is not a limiting factor. We welcome everyone who has something to do with Maharashtra and something to do with the Maharashtrian as a economy, Maharashtrian as a growth engine for the nation. And we have committed that we will contribute 1% of the GDP by 2030 as a community across globe. So that's about Garje Marathi Global. It's network of professionals giving selflessly to the community, giving selfless to people who want to learn from the decades of experience. Formally welcome to every one of you. Manojji, I'm going to hand over a baton to you now. And please take over this conversation. I'm really going to be looking forward for the conversation and the summary thereafter. So I understand maybe a bit of it from the non-science background. but still it's going to be a good experience for me. So Manojji, over to you. Welcome again. Thank you very much. I'm going to go on mute now.
Thank you, Madhavji. Good morning, good afternoon, good evening. Good. Thank you, everybody, for joining. I know it's a weekend and appreciate you taking the time. Madhavji gave an introduction about Garje Marathi and purpose of GMG Select Talk. So, given that background, we thought about the topics that is interest to entrepreneurs, professionals, students, and we thought the one topic that is not of interest, just of interest, but that touches everybody's life is health. And related to health is drug discovery and development, because who hasn't impacted by drug discovery, illness, wellness, and all that. So today we are talking about drug discovery and development. At the same time, all of us are hearing about AI, agentic AI, and other factors of AI, right? So these two becomes a focus point that we are all aware that drugs are always, the treatments are getting costly. We hear the side effects after many years, and sometimes the treatment takes years to come to the market, and they are delayed and all that, right? So there is one axis of we need more better drugs faster to the market, safer, cheaper. But at the same time, the reality is different. At the same other angle is AI is coming up, AI technology is evolving. There is a lot of promises of AI. There are also some risk of AI, or there are some shortcomings of AI. So today we are going to hear how drug discovery and development happens, because that's the first part, and where AI can help and where AI is still a limiting factor. So that's the context of it. I'm going to introduce our panel speakers. Dr. Bhushan, can you give a short introduction of yourself?
Yeah, thank you, Manoj. So my name is Bhushan Bonde. I had been in the pharma industry for the last 20 years. After my PhD in Oxford, I started serving for pharma, healthcare into pharmaceutical industry, and been working in field of machine learning and AI across from large to small, medium-sized pharma in United Kingdom, here in London and around areas, Oxford, Cambridge sector.
Thanks, Dr. Uday. Bhushan, Uday.
Yeah, hi. Thanks, everyone. Thanks, Madhav, Manoj, everyone, GMG. I'm long time association with Garje Marathi. So my name is Uday Khire. I live in Connecticut, very close to Yale University. I'm a medicinal chemist. After my PhD from National Chemical Lab, I moved to US. I did a couple of postdoctoral trainings. Then I worked for Bayer Pharmaceuticals in cancer discovery for about 11 years. And then I started my first company, and then we have been doing drug discovery and development. We had one company where we sold it out to another local public company, which are developing the clinical trials for the compound that we designed and discovered. So I'm really looking forward to the conversation today, and also communicating with everyone involved here. So thank you.
Thanks. So lastly, my quick introduction. I was born and brought up in Mumbai, moved to San Francisco Bay Area last 30 years, working in enterprise, AI, ML, not the medical pharmacy, but AI and ML in terms of enterprise reliability, accuracy, governance, delivery, safety, and all that. So I'll be moderating this panel. Before we go there, just a quick claim that, all the discussions they are happening here, these are our personal opinions of speaker, and these are not formal representation of our employees or institutes and all that. So this is a very informed topic, but it's just a personal topic. With that, I would suggest that since you've introduced yourself, do you mind giving a little bit details about your journey to where you are here, from your college, your company is formed, and your patents or something?
Miss, who's starting?
Uday, you starting? Yeah.
Oh, okay. So as I said, I mean, I got my PhD from the National Chemical Lab in organic chemistry. Then I did postdoctoral at SC Bound at Purdue, and also like UC Irvine. And then we worked at Bayer. So at Bayer is where actually I got to really apply the synthetic or medicinal chemistry that I learned all over the years. And we worked on the first project that was like a very new target, one of the kinases, which was at that time, like I'm talking about 98, 99, was very new. And we started from scratch. We developed a molecule, then we pushed it to clinical trials, which in 5 years became, about 5 to 6 years, became a drug. And I was just listening to our CEO that time, and he said when they were trying in clinical trials, some patients who were given like 3 months to 6 months to live, they were living 5 years after that. And that gave us the first, you were like, oh my God. So what we did in the lab now is actually helping patients to live longer. And that was the motivation that stayed with me till today. So I'm trying to keep doing the same thing. After our Bayer days, I started my first company, Chemin Pharma, in Connecticut again. So we worked mostly with Yale founders and worked with their startups, and then also some of the smaller companies in here and Boston area. And while doing that, we also developed a MEC. It's another target in the same RAS pathway. We developed a macrocyclic compound we designed. It's a very, very small team. I mean, if you see numbers on the clinical trials, like how much it cost, our cost was a very small fraction of the whole thing. So we almost developed IND stage. So the molecule was IND ready, and then we sold it to another company because at that time our expertise elapsed. I mean, at that time I didn't have much expertise in the clinical trials, so we decided to give it, sell it, and then they are taking it forward. Just it's looking pretty good at this point compared to what its competition is, and I'm very, very happy about that development. After that is done, what I decided, okay, maybe let me start something in India because that's where I came from. I know the city very well, so I founded a company, Chemin Pharma Products Private Limited in Pune. And that's where we are developing something called protein degradation. This is a new way of doing medchem. Instead of just forming like inhibitor, we are trying to destroy the protein from the body. So the protein is not there at all. So we're working on that. We have a team there. They're already working very good. So we'll hopefully be able to start testing soon and hopefully get the IP patent and move forward from there. So collected a lot of experience in drug discovery and development over the years, and really looking forward to discussing with. I'm so glad to have like Dr. Bonde and Manoj, and looking forward to hearing from his about his AI, the model, and how can they help me, because that is also something I'm considering we should be using with our company in Pune.
Thanks, Dr. Uday. Dr.
Yeah.
Bonde, you have a very interesting background. You are a professional. You are also in a college professor. So let's hear from you before we go into detail.
Thank you. And interesting background. Really impressed with your achievement so far, and good luck for the future as well. My background is really interdisciplinary. So my first degree is in pharmaceutical sciences as a pharmacy graduate, and then I changed my kind of career a little bit to join UDCT in chemical engineering, MTech, and that particular change, which is basically moving from pharmacy to chemical engineering, which is more of a mathematical heavy side, is what brought me to the AI and machine learning. So my first work back in India in 1998-99 was my thesis on neural network in drug discovery, where I was building a basically very small, shallow neural network when there was no deep learning concept developed in 98. The computers were Pentium 1, 2, 3. And so we didn't had GPUs available that time, even though GPUs did existed in time. But that was my model of working, on a CPU-based small cluster of computer, train a small model where we have that time hit, like Uday said, kinase was very hot in 98, 99. Silicoxib inhibitors, which is COX-2 inhibitors, very, very hot. So my first approach, my first thing was to model a COX-2 inhibitor, which is basically a best in class, even today, kind of a painkiller for painkiller. If you have a dental pain, then people will used to suggest nimesulide, which is a COX-2 inhibitor, silicoxib inhibitor, picoxib, and nimesulide. So that series of compound I modeled to build a first small neural network to predict if you give, let's say, pharmacokinetic property, physicochemical properties of a structure, then it will predict what will be the activity of the compound. And it was a very small, let's say, purpose-built neural network. it did little bit overfitted because of the lack of the real data series. We are only talking about 300 to 500 compounds that is used to train the model. Let's say 50 of them were used to break the model performance. But that was a journey. That was where I got introduced to the machine learning and AI as a practice. But then I moved on to do my PhD in Oxford, where let's say I did move a little bit in a medium time away from machine learning, but I did stayed let's say deep learning and AI, but still in machine learning, where I was simulating life on computer. So my PhD was simulating the cell on a computer. How can you perturb the cell on the computer and predict it? And that journey followed through my work in Sanger Genome after my PhD, working on some genomic profiles, identifying cardiogenic profiles of patients using machine learning models to see why cardiac arrest occurs in certain people, using the genetic profiling, RNA-seq, RNA and DNAs, and see what is the result. To the pharma companies, then I joined GSK for 7 years. Then I worked in UCB Pharma as a medium size, Belgian pharmaceutical company, where I was building all the deep learning models for scientists in the lab. So democratizing the AI was a kind of a concept that I had been serving to pharma industry. And recently with Evotech Pharma for last 5 years, I had been practicing these to, let's say, deploy the foundational models to the industry at scale. And that is the experience I kind of accumulated over the 20 years for my industry. So where we are in this in the journey, I am still I'm a learner in the realm of the AI, because this field in the AI is moving so rapidly. Just 10 years ago, when this deep learning let's say phenomena started, there were models now that we cannot imagine that could be possible, and we will discuss some of this model today as we talk in our discussion. But yeah, here we are, and looking forward for a good discussion. Thank you.
Thanks, Dr. Bhushan. So before we go into the deep dive, right, I think we just need to speed up a little bit. So now that we have heard from our two panelists, right, so drug discovery, basically, as we mentioned, there is a discovery phase and there is a development phase. As we'll go through this, the webinar, we'll see that in the discovery phase, there is a multiple areas, and also in the development phase, there are phases of clinical trials and other part. So we'll briefly touch what is drug discovery and discovery matters, and then we want to go to the phases, the preclinical phase 1, phase 2, phase 3, phase 4, phase 5. So Bhushan, maybe you can just quickly go through this, and Uday will quickly go through this, and then we'll start on the next part, which is the preclinical and the phases.
Surely, yeah. I'll briefly touch it, and then I'll let Uday go into the detailed phases. Basically, drug discovery is a process where we want to identify a chemical substance. Could be a protein, could be any entity that could basically cure a condition, a physiological condition that is happening in the patient, which is, let's call it, not normal for a given patient. And so here the word I'm using, let's call it normal, because defined normal is one of the big debatable topic, which is not something we can discuss today. But when we say normal, is a bit debatable. So we have to always say that with respect to a condition, define normal, and so that normal. But again, coming back to the drug discovery processes, here the key substance, key thing we are looking for is the substance which we are taking as treating a disease should be very safe, efficacious. That means it should have effect when it's taken, and it should be selective, and it has manufacturability. It should not be degrading by the time it is coming to the market or coming to the shelf life. And so these are the key stages that we need to maintain throughout the drug discovery process. And why it matters is because as you mentioned, there are millions of patients that still do not have therapies. There are around 16,000 rare diseases exist, and there are more than 10,000 common diseases. So there are 26 around known phenotypic conditions that we have. And believe me, you will be surprised to see more than 50 to 60% doesn't have any treatment available today whatsoever. And so if you can see the scale of a patient needing some kind of a treatment is much more really a need of a time and need of the hour for today. And so again, when it comes to pandemics like COVID-19, we saw how much is the whole world had costed it as a cost of treating this pandemic. We really need to be ahead in the game to avoid such a pandemic happening, so that the cost implications around the whole world as a earth, I'm saying, not as a country, can be ready to face the challenges arise because of such pandemics. And then ahead, as we see, as I remember Uday just mentioned that patient has been living now from 1 year to 5 years for any disease which has, let's say, even for cancer. AIDS, take an example of AIDS. In 1980s, there was no treatment available. Today, AIDS is almost 99% curable, in a sense that it will be still there in the person's body as a fraction, but the person can live healthy life for rest of his life and will not have any problems. And so that is the achievement that we had been done in last 20 years or last, let's say, a few decades in pharma healthcare, that can be really saving lives in miraculously for patients. And so I'll hand it over to Uday for further points to add.
No,
Thanks.
I mean, you explained it very well. So basically, it's so important to have drug discovery continue because, as you said, there are so many patients with unmet need. And it's more important now because a lot of breakthroughs in biology are happening. And those breakthroughs in biology will be complemented by good chemistry and good PKD and good science, and can be actually reality in terms of treatment options, right? For example, what they call is CRISPR. That was a big breakthrough. How you repair the
CRISPR-Cas9.
gene, right? How do you use that in discovery? Or the biggest story in last couple of years is GLP-1 drugs, right? It started as obesity or a diabetes target, type 2 diabetes target, right? And then it turned into people saw that people with this, they're losing weight. And now it's big market, obesity market. And because of this development, Lilly was the first company, became basically $1 trillion company in pharma. That was unheard before. So all these things are so critical and so important. So we'll go over this, but I think this is a very critical, very exciting time for drug development and discovery. And you will see with examples as we move on.
Yeah, thanks. So I think one of the things that people who are not close to the the biology and the chemistry, we still hear that, hey, there was a phase 1 failure, or FDA withdrew it from the market, or phase 3 became negative and there were people impacted. So as we understand, there are phases of drug discovery and development, and most of our session from going forward will be focused on these areas, where what we'll share is what are the phases, which people are involved, and how these phases are managed, what is the cost, what are the risks involved in that part. So with that, Uday, you want to quickly go through this and deep dive into this part?
Yeah, I can do that. Yeah, slideshow, please. Yeah. Yeah, so basically the whole drug discovery and development up to marketing is divided into two main categories. One is preclinical and then the clinical trials. And then after clinical trials are done, then marketing. If you're lucky enough to get it approved through FDA, right? So within preclinical, there are two important stages: basic research, and until you identify a candidate, a compound, a drug molecule that you want to develop further. And then you do non-clinical studies. That means like toxicology studies, doing studies in animals. For non-clinical, you need two species. Normally use like rat and then dog. But if dog is cannot be used, then people go for monkeys. So this is non-clinical studies. And based on that data, FDA, in terms of US, FDA will approve whether you can take this drug into humans or not. To get to the non-clinical, you have to have molecule, right? You have to have some new novel targets. So that most of the time comes from basic research, from academic labs, right? They come up with some screening cascade. They say, okay, this is the cell signaling pathway. This is RAS pathway. This is very important in oncology because about 30% of the cancers, this pathway is overexpressed. And there are a lot of mutations in particular pathway or in particular gene target. That mutations are causing a lot of issues. For example, there is pancreatic. If you take a pancreatic cancer, right? It's a very aggressive tumor, and survival is very difficult. I mean, the biggest example was Steve Jobs in pancreas. He died after knowing, even though it was identified early, but still he died within a year or so. So what I'm saying is, basic research is critical, and coming up with good targets, finding the meaning of that into actual patients is important. In India, the cancer rates are growing so fast. Uh, there is a lot of diabetes, there is a lot of cardiovascular. So that's where the basic research helps, and then the non-clinical. But once you have identified a compound, then we go into clinical trials, right?
So
So there
Uday,
are three
before we go
phases
into
of
clinical trials, one question, right? So is there like a ballpark success rate or dollar amount that you can tell us that it takes so much dollars, or it takes a success rate of 5,
about
10, 15%?
the success rate is about 15%, 20%. To get like approval from FDA to continue the clinical trials in humans, and the cost is like I would say about 20 to 50 million dollars to get to that stage.
Okay.
Yeah, it's nothing here is cheap, right? It's very expensive. It's time consuming. It's very expensive, but you'll see, right? At the end, it matters a lot. So in clinical studies, we have three phases: phase one, phase two, phase three. So once you have data from animals, tox data, your efficacy data, then you go to phase one in humans. It depends on the what therapeutic area you're working on. For example, if you're working on oncology, then normally you recruit patients with already having cancers or having tried other drugs and failed drugs, right? Those are the patients you recruit for oncology. But the other diseases, cardiovascular, diabetes, everything, you go for healthy patients. You have to show that in healthy patients the drug is safe. And then we have to also find a dose where which is therapeutic dose. That means at that dose you will expect the drug to work, and so your idea is there has to be a difference between therapeutic dose and a safety dose. So your safety dose, for example, obesity, has to be much higher compared to the therapeutic dose, right? So that you will have a good safety margin. So phase one is confirmation of safety and side effects. The most success rate here is about 70%. And the drug failed because you don't get that exposure. For example, if it's oral drug, right? It doesn't get absorbed, or it gets metabolized faster, so it gets excreted very fast. So there are a lot of issues with that. But I think success rate is pretty good because you have all the all the data from the animals when you go into phase one. Once you have phase one data, okay, you say, okay, I know that at 10 milligram, I can be effective. And at 10 milligram, I don't see much side effects or toxicity. It's not like a dose limited toxicity. So you go to phase two with that, and then you select a lot more patients, right? In phase one, you have about 20, 30 patients that you go to a much, compared to phase one, larger patients.
So
And
how
then
do you select this population? Do you select geographically different? Do you select age different? Because any experiment, this is basically an experiment, and creating a representative population in terms of healthiness, age is different. So could you share a bit on that part so that we are aware?
Yeah, absolutely. So this again depends on like what therapeutic area you are working on, right? For example, obesity, you can find so many patients, right? Globally, obesity is rampant, right? So you'll find so many patients. But if it is a rare disease, right? For example, it's NF1, neurofibromatosis 1. So there are only maybe 1 in 3,000 babies has the NF1 mutations, right? So you have to go and hunt for those patients all over the place, wherever you can get, because by rare disease, that means it's by definition, you don't have many patients. So you need to really identify and find those patients and recruit them for your clinical trial. For again, for cancer, you start phase one with already identified patient population. The criteria there is what is your mechanism of your drug, right? If it works on particular mutation, then you need to identify the patients who have that particular mutations, right? Because your success rate will be higher if you find the right patient population for that right drug. So it's like depending on the disease mechanism, lot of things. But in general, you try to go with male, female patients, and then for pediatric, you cannot go right away. Once you have established good drug, good benefit in adults, and then you know what is the level of drug. Like for example, 500 milligram. If you're giving a 500 milligram a dose at adults to get an effect, then at that point, FDA says, okay, now you can go to pediatric, but you have to go maybe like 30% of that dose. So you again start with a pediatric trial at 30% of those because the tolerance with pediatric and adults is different, right? So it all depends on, but male and female, they try to get every geographical, every location as much as possible. So they can say this drug is not only for Caucasians or Asians or for African Americans, so it can work everywhere. But having said that, there are some drugs which work really well in one of the like Caucasians. They don't work or only work in African Americans more because they see the prevalence of the disease or that particular mutations there. So it's a lot of studies, a lot of thinking goes into recruiting before we recruit the patients.
So one question I'm going to ask Dr. Bhushan, and before that, right, we see the success rate drops in phase 2 and of course phase 3. So Dr. Bhushan, you have done a lot of work on Gen AI, AlphaFold. Can you tell something about where these models are helpful in discovery phase or the early phases? Any thoughts from you?
Yeah. So AI can be applicable to multiple phases. Not each and every phase can be applicable. But like you mentioned, you took a very good example where AI has been making an impact. For example, the early phase in the drug discovery, where identifying right target and right disease biology, right protein, which can be a target. For GLP-1 is a protein basically that we are targeting, and so. In our body, around 21,000 proteins, as we expect, come from 22,000 genes. Around a proteome is formed. So we know each and every gene sequence that is contributing to protein, but there are many proteins because of the alternative splicing. So that number can go to almost 100,000 proteins in the body, giving from this 20,000 coding DNAs. And so some of them, it took years, almost like 10 years, to get a protein structure identified so that we can actually take a drug and bound to that protein structure. And so it involves a lot of rigorous crystallography studies, extracrystallography studies, NMR crystal structure identification, and all. Sometimes it is not even possible to get the protein into a crystal form because it is not soluble or not possible to get that. And so what AlphaFold and CoFold and the other model has been solved is that we knew from the known protein since last, let's say, 50 years of extracrystallography studies has been done since the early 70s, since the NMR and crystallography studies has been performed. We have around 250,000 protein structures synthesized today. Let's do a deep model on it, and that's called AlphaFold. The Google's DeepMind has been able to train on all these protein structures and able to create a deep learning model. And that deep learning model now means that as long as you have a coding DNA or a protein sequence, we call it, expression, kind of a sequence of a protein in the form of, let's say, beads, we will be able to predict how will be the protein is getting folded in a body or in active forms. And that active form means that we are very easy now to identify the right targets, or right small molecules, or even the other alternative modalities that can bind to that protein. Could be RNAs, as Uday mentioned. There are other TPD, like protein target degradations. Therapies are coming, which we can take it. But to do that linking, removing that protein, we need to have that structure where it can bind to that. And that was one of the biggest challenge pharmaceutical industry had since last few years, which has been now solved by co-folding models. And so what we have is that a plethora of co-folding models, which we'll see later in one of the slides, we have compared, is how can we deploy these models internally so that we can get decision making clear. Clearly, I mean, today, if you have a protein of hypothetical protein, you can synthesize in the lab. You don't need to wait for crystallography structures, etc., which is years of FTEs. Multiple people work on it, and so that can accelerate your timeline from, let's say, years of work to, let's say, a month. Within a month, you can actually validate that in the lab. And so these are the real foundations that have been happening in the clinical trial. Again, it's not directly playing a role, because in the clinical trial, by the time you're doing in the patient population, you already have identified bounding. But what is surely going to affect is that the better in the biology selected in the early phases, you have a less chance of your clinical trial failing in phase 1, phase 2, phase 3. And that is the promise of the AI model in the clinical trials.
Thank you, Bhushan. Uday, just please continue with your phase 2 and phase 3 and explain us what are the challenges there. I think phase 2 and phase 3 is the biggest, costliest, and seems to be the more critical. So please shed some light on that part.
Phase 3 would be the extremely costliest, right? It could go up to $1 billion expense. And so phase 3, of course, you have much larger population. You follow for safety, efficacy, everything. If the drug really looks good, you have to continue interacting with FDA, like all the regulatory agency where you are, right? And then basically convince them that this drug is working. And if there is some other like first line therapies available for that particular disease, then you have to show that this is better than that, right? Not only, oh, okay, this is also good, that works, this is also works, that is not good enough. You have to show that this is better than whatever is the current therapy. And then once you collect all the data, you apply FDA for the new NDA that called the new drug application. It gets reviewed. There is a committee that basically formed for that particular project, and they tell suggest FDA that this you should approve or not, or whatever it is. And sometimes FDA also, like 90% of the time they go with their recommendations, but sometimes they don't. Sometimes this even they have said no, don't take it. FDA has approved, and even the other way around. And then once it's approved, right, then you're ready for launch. You're excited, oh, new drug, your stock jumps, everything. If you're a smaller company, you are so high. But then once that is approved, then the work is not done. Even if it is approved, you go to the phase 4, what they call. So after approval, because even phase 3, there are a lot of patients. Phase 4, when it's approved, it's expand tremendously, right? I mean, if you have 1,000 patients, it will be like millions, 5 million, 10 million everywhere. And then what you have not seen in terms of side effects in clinical trials, sometimes they pop up after it is approved. And there are examples of drug which were withdrawn after phase 4, after in market, after being good drug, popular drug, and then they say, oh, no, 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 these guys are developing some type of rare cancers. And that shows up after 10 years of treatment. Of course, you have to withdraw it. You have no choice, right? And a lot of issues happen. So phase 4 is equally important, even the phase 3s are the critical to get the drug approved from FDA. And then
There is
marketing
a
and all that.
question, Uday, there is a question from one of our other experts in GMG, Anita, about ADC and CAR. Are we touching ADC and CAR? So I'll leave it to any of you to take it.
We can touch ADC and CAR because those are, again, I was talking about the breakthroughs in biology, right? So those are the things that are so critical. CAR T cells is basically, if it's mostly used in cancers. If you have cells, right? We have our cells, they have fighting mechanism. So if there is always this, there is a cell signaling where there's tells cells to start growing, then there are other cells that say, okay, we have enough, stop it, kill it. But if that cell which says kill it is not working, right? So it basically says it's not functioning because there is some mutation that needs to be repaired, right? So what people are doing, they're taking that patient's cells out, they repair it in vitro in the lab, and then putting it back into the patient. They start working. It's amazing. When it works so beautifully. But there are, of course, issues which will get resolved as we move on. And ADC is antibody drug conjugates. So basically, you take a drug, but it goes to all tissues. It gets distributed everywhere. And a lot of toxicity could come because of that. You don't need to go to heart. You don't need to go to lung, right? So if you have lung tumor, then you want that drug to go to the lung. So there is antibodies that are expressed on the surface of the lung tumors. So you put that receptors on the drug molecule, conjugate that. So that basically go and bind to that lung tumor without going anywhere else. And it gets internalized and release that drug inside that tumor. So these are very exciting happenings in the drug discovery, along with, as I was saying, like protein degradation, right? Those are very important because traditional medicinal chemistry, the protein will be still there. We just used to block it from functioning. So the protein will be there. But now we have found body's machinery, our own machinery, to kill that protein. So basically, they wipe up from the body. So unless they resynthesize back, they will be gone. And that biology is different. There's only just recently first drug got approved which uses that mechanism. So yeah, I mean these are the very, very critical important developments.
Thanks, Uday. So I'm going to take one more question from the chat and then we'll continue with our discussion so that we can finish the discussion, the panelists, and time allowing, we take another one. So this is from Praful Nabar. I'll give it to Bhushan. So he's asking, is the speed of data crunching the main contribution of AI here, or is there some intelligence involved? So
Yeah.
Dr. Bhushan, just give it some quick thought, but we have a lot of
Yeah.
content remaining. So
Sure,
let's be
sure.
aware of the time as well.
No, it's a very good question. So one thing is sure, the data crunching is one point in AI which is helping. No one single person can hold the knowledge about any drug or multiple, let's say safety aspect of everything into their memory, and that is a problem. I mean, drug discovery, you saw how difficult it is, how many stages involved, and the chevron which looks linear is not linear. You have like a spaghetti-like decision making involved, and so no one person or no one even talented group of scientists can hold the whole knowledge needed for drug discovery into one context. While the AI model can hold that knowledge in one place and can be able to give you better intelligence based on all aspects involved in the drug discovery. And that is what we saw in the current models like AlphaFold and all. Why would AlphaFold will behave better than a protein scientist who is working, let's say, 50 years in identifying the protein structure? Because he will not be able to just looking at a sequence, will not be able to apply the knowledge saying, oh yeah, I have seen this in common into, let's say, 15 proteins in a family of proteins. And so I can put that in here and I can make the structure. That is exactly the struggling. So yes, there is a data crunching is contributing, but along with that, how to apply that correctly with the reference to the problem you have is also the intelligence is also brought it in by itself. And that is what really helping in the AI based model. And we'll see also another point which is leading to the discussion, and I will ask you, Manoj, to remember Agentic AI, which is what exactly this point is leading to. The intelligence until now, so the workflows in pharma company were very well known. Dr. Uday will know there are 9 workflows where you can just say, take 100 compounds, screen it overnight, and in the morning you will have a list of compounds available. That was the dumb workflow, just doing in kind of a machine learning check, whether this particular structure had the toxicity or not, whether this ADC has this toxicity or not. And that was the way to kind of do drug discovery. With the agentic AI now, each agent is independent. It can run a cyclic way when it can do loop over until it identifies the intelligence, knowledge it can extract from the model, and then it will associate, it will link that to a knowledge. On the screen, if you see, it is too small, but on the screen, you can see that this is an example of agents working in pharma company to take a decision. And each of these agent is an independent actor which has a knowledge of a model, AI model behind the scene, to understand what is going on. So for example, there is a lab agent which will go to the lab and fetch some data or fetch some experimental analysis. There is an agent which is gets a molecular information from smiles. So it will get a toxicity from smile, get some activity from smile, get some bond length from the smiles, something like that, bond angles on and Lipinski's rule of 5 identified from the molecule, so which is basically identified for druggability. And so each agent is independent and it will keep doing until it's satisfied that. And there is a supervisor agent which will make sure that none of this agent goes into the chaotic systems. So that is the intelligence we are talking about. Guardrail agent will make sure that you are not crossing the limit of what we say safe limit of working. That means you are only responsible for fetching the information that is asked for, and not something like 2 is equal to 4 or some kind of write me a song or something. That is a responsibility of a guardrail agent, that the prompt is going to say stay what it is and not do the other task in the thing. And so this is exactly working concept in pharma company. That is a paper of those who are interested should read, that accelerating drug discovery through agentic AI by Felice et al. Published in 2025 with the examples of AI agents. And so I hope that answers your question, Praful. Thank you.
Thanks, Bhushan. And quickly, right, agentic AI, right, as Bhushan said, it's not just the speed of data crunching, it's the inference. It's digesting lot of knowledge across vast domains, vast documents, vast archives, and doing the inference, right? Although I don't work in this, the agentic AI is influencing all areas. So like today we have agents that can run for hours, they can run in parallel, and they can coordinate and summarize their knowledge. And briefly,
Yeah.
Dr. Bhushan, what you said is there is something called adversarial review. So once
Correct.
AI comes up with a solution, it's not that you just take AI for granted. You can also ask AI to run an adversarial role, review it from a skeptic perspective, review it, say, hey, this
Yeah.
is something going wrong. So if you want to take this message, right, a lot of things you hear in the market, they are just kind of headlines, but there is a way to use AI which is, leverage the skills as well as tries to implement the guardrail and the adversarial review or the guardrails. There are ways to put that part, and although it is not perfect, it's getting much better into that part. So thank you, Bhushan. So we are going to
Thank you.
continue with the phase 3 and phase 4 part of it briefly, and then we'll go to the model. So Uday, you want to briefly wrap up phase 3 and phase 4?
Yeah, no, basically, I think that's it. I mean, we are done up to phase 3 and then phase 4. So phase 3 could be very expensive. Again, depending on what therapeutic area you are working on. If it is obesity, diabetes, it's a very large trial. Alzheimer's, CNS, right? They are very long trials, very expensive trials. That's why smaller companies shy away from that. But if it is rare disease, if it is very small population oncology, they could be very quick, and then they would be costing less than 500 million. So, yeah, it's a very, very exciting journey, right from the discovery all the way to the market approval. I'm looking at the time, and I think I don't want to go my details here.
Yeah. So we'll hold on the question. We have all the questions. We have the contacts for all three of us panelists, so that we can't answer any questions, please get back to us. Uh, we'll take one question. Mrunalini, you have a hand raised, so please unmute yourself and state your question briefly.
Okay. Thank you. This is a fantastic discussion, and I am also trained as a data scientist 10 years ago. Dr. Bonde, I mean, it's really fascinating to hear from you, the pioneer of AI in the drug discovery. So regarding accelerated drug discovery, there are many, industry, private, I mean, the public-private partnership coming up, which is something like Transcelerate. And they are doing, somebody posted also acquisition of in silico. So
Correct.
how we use the generative AI into in silico trials? Basically, that is where it has more potential, I believe, uh, because those would be AI driven rather than, at least up to safety. Phase 1 trials can be in silico trials. That is, I mean, can be, started, explored first. And also, I would like to know now, AI and genomics, I mean, still there, evolving, but still, like I would say yesterday or today morning. But now, this time, it is generative AI and pharmaceuticals, I would say. So, how we can leverage AI into these trials to accelerate these kind of trials? Thank you.
Sorry, I missed your first question. Could you repeat your first part? It wasn't
First
clear.
part was like in silico trials, how we can leverage that, and do you have any, because in silico is mostly working on that, and that is why that acquisition, because that is the question also somebody asked.
So in silico is a big company. I mean, they had been doing it for last like 5 to 8 years. I know Alex very well. He's one of kind of a friend. And they had some 3 products. I don't remember the top of my head, but they had like one for small chemistry, AI-based small chemistry workflow development, where they had been doing their own internal AI model development. They also had retrosynthesis like small molecule synthesis, and the bioformable, where they can do assay development and assay identification. And they did not only train the AI model, but they also had a lab validation done for that. Because if you see in Silico's website, they had their own targets, which is in like say phase 1 to certain phase 2, phase 3 in progress, and then that has been acquired by Eli Lilly. I think when they were acquired, they said they will be able to independently work, but still Eli Lilly will also use their workflows and internal pipelines. So the acquisition was more about let's say, acquiring the ready-made pipelines for Eli Lilly's internal tool sets, internal drug discovery programs to accelerate. That was, I think, what my knowledge is. But I could be wrong. And the second questions, I think you had asked for how can we accelerate these processes. Is that correct?
RLT. I mean, this radio pharmaceutical, how we can in this, because these are very very nascent stage. So how AI
Correct.
can be leveraged in that type.
So unless we build some foundational model based on the known data, radioactive toxicity data, etc., I mean, somebody has to do that work, correct? I mean, AlphaFold wasn't true because couldn't be built by any of the academicians, because it needs to come up with all the right, let's say, environment there. Only Google had a power to burn such a big farm of GPUs. I mean, at least thousands of GPUs burned for a year to get that model, fine-tuned, etc. So that kind of ability of engineering, they have engineering in terms of AI to do it. Even for a big pharma, the cost of building such a model is very huge. So my point is that to do something like, let's say, toxicity prediction for a radioactive or radio pharma, etc., we need to have the foundational model trained on the data. And if the data set is not there, we need to willing to at least do some lab experiment and get the data at least to the form to train the models. Uday, anything you want to add?
No. I mean, that's very good. I mean, you said it well.
So let's continue with our content. So we have still more content to present. Appreciate you all taking the hands. Please put your questions on the chat. We are also going to share our contacts. So in the unfortunate event that we cannot finish the kind of questions, we'll get back to you, or you can reach out to us. So we want to touch quickly on the AI part of it. And Bhushan, if you can just walk through it, and Uday, maybe you can chime through it for next few minutes on that part, so that we can still be time sensitive. It's getting late in India, and then we can wrap up.
So this slide will be anyway shared in the follow-up, let's say, video and all. But so I'll not take too much time in the worry of time. But what I want to say that AlphaFold was the kind of program which has game changer. There was 40 years of belief. In fact, there was a scientific paper called Leventhal who proved that it is impossible for any computer model to predict the structure of protein. He gave a mathematical lemma, by the way, not just a Leventhal's paradigm, they call it, is that it will take end of the world to predict each conformation of a protein, because it will be 2 raised to n minus 2. The n is the number of amino acids in the protein, to identify the structural conformations by mathematical an algorithm. So that's why he said it will be impossible to do such a thing in the humanity in the world. But that kind of Leventhal's paradigm was proved wrong by AlphaFold by training the deep learning model called AlphaFold 2, where you just take the structure of the sequence of the amino acid. It's like a bead, and then it will be able to fold, and they did very elegantly. The core idea was very simple. You do multiple sequence alignment with the known proteins, and then it will generate a guided template. So it will generate few small templates, and they join the templates. It's like a jigsaw puzzle where you have a puzzle fitting into each other, and you will get the whole story together. And so then it started with the X-fold, a number of fold. ESM fold came from Meta, which is a competitor to Google. Then we have Chai and Bolds, and then there are other models which were based on the ESM folds original data. If you see Chai and Bolds, they are really using ESM folds open source model. They just kind of fine-tuned it to do work on RNAs and DNAs and other kind of modalities. But the key thing is that no one model is going to give you everything what you need. And in the real life, you need to ensemble of models depending on what questions you are asking. Are we asking protein structure? Go to AlphaFold 3. Maybe for academicians, go to AlphaFold 3, because only academicians are allowed to use AlphaFold 3, and that will give you better prediction. If you want to scale and speed, you go to ESM fold. You will have some areas of the protein which will not be correctly rendered, but you will get ballpark right. If you want to predict a complex modeling of say kind of a compound, how is binding RNAs, how is binding to the protein, of which you folded by using AlphaFold, then you use Chai1 or Chai2 now. There is a version 2 available model. And the Boltz 2 version is where you want to identify affinity, because affinity is what exactly in drug discovery identifies how the drug is binding, the key PK association dissociation constant, and that could be available using the Boltz model directly to you. So you don't only get the structure predicted, but you also get the bound structure where the chemical or the RNA or an entity that will be, or even another protein in an antibody. I think Anita madam asked, like, are we going to discuss ADC? I'm not an expert in the ADC and thing, but Boltz model could be able to predict how the proteins and the domains are actually binding with each other. Protein-protein interactions are happening, and they could be able to give you that kind of predictions. So this is the summary of that depending on the model, you need to ensemble it. You sometimes have to use more than 2 models to get a confidence. And
So
Uday, maybe over to you.
From your perspective, right, you have looked at the molecules, you have your own company. Rather than saying, hey, this is all good, share us your perspective where you still see the gaps, where you still the risk, and where you would like the AI community or AI technology company or AI to improve. So give us a skeptic perspective, skeptic with a positive feedback that, hey, look at this part.
Yeah, I still
So
want
that we walk away with a balanced perspective, not
I
one
want Bhushan to be my
other.
friend. So no, it's I think what Bhushan explained so well, right? I mean, basically these are models in development or some are really developed. AlphaFold 2 also, I think if you already have a good X-ray structure of your ligand bound to the protein, then also AlphaFold can explain like what particular mutations in that receptor could affect your binding or not, right? If the drug can become ineffective based on that. So that information could be very, very useful. Having said that, I mean, this whole modeling or like even prediction about solubility, prediction about binding, all is really helping, but still we have to go to lab and make the compounds and get tested. And I would like to see like a real AI drug approved in market, right? That's not happened yet. It's still in early stages. And I think all the predictions help a lot, but still we have to do it in trials, clinical trials. We have to do it in lab. We have to again go to in vitro biology. We have to see the efficacy or affinity. We have to see the toxicity actually in animals and in humans, right? I mean, that you cannot escape from
So
that.
where is
Nobody's going to
one
approve a
area?
drug based on AI prediction. That's what my only.
Where is one area you think AI is lagging much behind in your perspective?
I would take the other way around. And I said AI could be useful really in terms of what called the drug purposing. That could have really good value because if you take a patient, directly go to patient, every patient is different. There is something called personalized medicine, right? That is developing hugely. And every patient, every cancer is different, right? You can say, oh, this guy has a kidney cancer or liver cancer. Within liver cancer or there are different mutations. So same drug would not help to particular that. So AI can predict, okay, for this particular patient, there are this numerous drugs or molecules already available. You can treat this combination, or even if it is not drug, just went to the clinical trial, and they say, okay, this molecule could be very useful for this, and then people can take that and try in vitro for that particular patient, right? They can take the patient's cells and then see in vitro is working or not, and then really improve the treatment rather than just giving the standard of care. I think that's where the real applications of AI, from my perspective, would be very, very critical, very important.
Thank you. So I think we talked about AI area, but I think the proof of the technology works or not is whether it has improved anything, right? So Bhushan, right? You use a lot of AI. We discussed some of the KPIs. So what is the stuff you shared this with our information? Please tell us the audience, right? How would they measure whether it's a AI, whether it's a technology A or B?
Yeah.
Ultimately, it's a number game. Can I see the game?
Correct, yeah.
Can I see the performance? So please walk with this?
And there is a question by MV Peshwa, I guess, on the things like what is driving this valuation. So, I mean, in the one of the ultimate things to answer is that the success KPI. Any project I lead in the industry, I need to show that deploying this project has able to show the impact, whether it is an AI project or not. And this is what I think we do in real time, that if the AI project, let's say AlphaFold model, folding the model internally in the organization, if you are able to say your cycle time for predicting protein structure has been increased by 20 to 50% compared to what it was earlier, so you have to do X-ray crystallography and all, then you measure from weeks to how many weeks it took from that target ID to lead optimization. If it has reduced by 50 to 20%, 20 to 50%, then obviously you hit the success KPI. Then scientist satisfaction. In the structural biology department, you can ask monthly survey and say, did it really help you? Did it really made the difference for you to daily life? And then based on that, you can do it. But the key thing is to remember is your pipeline really moved ahead? Have you seen the increase in your pipeline? The pipeline is where you say compound, how many compounds are in phase 1, phase 2, phase 3 in the market to come. And this is basically most of the pharma companies today facing the weak pipeline in the industry. And if that is moved, then we surely say this is really working, in the healthcare. And that is how we successfully measure the KPIs.
So we'll take one question from the chat. Unfortunately, because of time, we have to be sensitive. We'll skip the valuation part because that's kind of sensitive, like subjective and stock market on that. So there is a question about Shubhankar Manjrekar. I think this is a good question. The question that as the agentic AI shortens the timeline, does it take the competitive advantage away from biopharma, or is the AI still co-pilot? It's not going independent.
Yeah.
And I think that will kind of start to closing down.
Yeah, I mean, this is a very good question. What we saw AI for 10 years ago, agentic AI is a baby in the market now, just 1 or 2 year old. So there was no MCP server, by the way, before 2025. If you remember, December 2024, first my MCP agentic AI server was launched in the thing. The open clock came in January 2026, and that kind of brought in the kind of agentic AI. So most of the pharma companies are now setting what was in the olden days, the workflows I showed, 9 workflows, pipeline pilot workflows, there are some drug discovery workflows. They were drug discovery workflows. They were just doing dumb kind of, analysis. Now, most of the pharmas are now converting that workflows into, by agentic enablement, into some kind of, independent decision-making workflows. It's early days. Again, this is really important to know that doesn't mean you will succeed. The success will depend on how the human, let's say, governance on this thing, how surely your scientists are agreeing with the agent for performing and analyzing the agent. And then there are also, as you mentioned, Manoj, you said you can actually competitively identify the output of the agents, saying, let's challenge, let's criticize. And so these critical pathways are now set up by most of the pharma companies. Some companies are ahead, some companies are setting, some companies are trying to deploy this in real time. So the success will be surely visible in next 2-3 years. And as I said, there is no doubt that it will kind of, so this is where it is leading in one sentence is, this is where the pharma companies are saying a digital twin scientist is going to work in the pharmaceutical drug discovery pipeline. That's full stop. It is inevitable, whether we like it or not, whether I like it or not, whether I want to see that is not, it's not my choice. Okay. This is forced on us. And this is what the digital twin scientists in pharma will be working for us. How it will succeed is depend on how the models capable of powering the agents are getting progressed, how their intellectual is really getting day-to-day better. Surely they hallucinate. Surely there are guardrails in place. Surely the guardrails need to be tightened. Surely there are supervisor agents need to be tightened. But the real thing is that the capability of these intellectual agents will be driven by the models that are really burning. Today we are in a spree of burning tokens. $1 million token can be within one, let's say, year could be burned. Are they bringing the value? Are we really burning those tokens sensibly? Are we really be ethical in burning? Are we really saying the value out of them? That is what is going to drive the pharma drug discovery. The hype is at one place. The value driven is another place.
So
Yeah.
go ahead.
Yeah.
Go ahead with that.
So
Please,
Manoj,
Uday.
you talk about competition, right? I mean, just one question I was thinking, like, if the AI or the compound or the computer throws you a molecule, right? Just a structure, nobody chemist thought about it, and then you develop it becomes a drug. I don't know how the patenting will work for that. I mean, maybe Bhushan, you can answer, because normally the chemist who comes with that, the structure, right? I mean, they are all co-inventors of that particular patent, and that is a very critical patent, composition of matter of that. So if Pfizer comes with the same, the model you developed, right? Pfizer comes with the same drug, Lilly comes with the same drug, somebody else comes with the, right? So it's very difficult. I mean, that will be question down the road.
That will be question. And you hit a very nail. At the moment today, there are 5 models: Opus, GPT-5, Gemini 3. Everybody is using same model. So everybody is getting the same or more response.
Exactly.
Everybody is creating the same or more IP. Who is going to decide whose IP is right?
Exactly.
And that is really the worth looking into question. And I personally don't have a question, even though I'm a panelist. I'll be very honest. This is too early to talk. The laws are kicking in. The laws are now catching up with the AI models. There are really strict EU laws. By the way, 2025 started. It has a flaw. It started with goodwill, but the EU AI law allowed drug discovery to be kind of exception, because it's for betterment of the thing, to keep away from the AI deployment law. So if the model does a mistake, the responsible person who had developed a model will be held responsible. But it doesn't still kick in into healthcare industry, because that was kept away from that particular strict laws. It will come. It will eventually catch up. When it will be, maybe to next year, maybe in next 6 months, I cannot tell.
Yeah, and
Thank
from
you.
my perspective of using agentic AI, right? As the model's capacity grow, maybe some of the work will be threatened, but the productivity gains are so much that the other jobs that were spending years on menial work or data collection, not only they will become productive, but a new category of jobs is going to emerge, whether in drug discovery, whether in other parts of it, right? Whether it is testing of part and all that, because you can do so much groundwork that there will be a lot of other parts that will be brought up.
Yeah, agree.
So with that part, we are close to it. I know we have some lot of great questions. What I want to do is I want to give our panelists, if you want the team to walk away, or if you want the audience to walk away with one last thought, what's your thought? Would I start with that?
Yeah, so we just discussed, right, a lot of discoveries. So the current where the pharma research and discovery is very exciting. And as Bhushan explained, the AI can help those developments, right? So this is a good time to jump into the pharma careers. And that's what I would say, like there is a lot of excitement ahead, and we'll keep watch. I mean, next 10 years or next decade or so, or 2, I think we will conquer a lot of diseases that untreated totally, completely right now.
Yeah. And I totally kind of agree with Uday. From my side, attention is all about you care about. So if you want to be into this pharma AI kind of journey, as Uday said, it's very interesting for next 10 years. Be sure to make sure that you pay attention to the details, because as we use the models, and you will see that models hallucinate, if you miss your attention of what you are looking from the model, and then model can give you a really gibberish result, you are actually doing much worse than getting help from the model. So please pay attention to the details when you are talking about AI. And my another kind of quick one sentence thing is the current problem in any pharma, doesn't matter what pharma you are, is the development of the drug from the initiation, the discovery to the market is 10 to 15 years, and it costs, let's say, almost $1 billion worth of economy. It is not survivable. You have a lot of unmet needs. So even if the AI halves this distance, I don't want to say that let's do like a COVID drug discovery in 1 year, but even if you halve it, the cost of the burden cost on everybody, the AI, the agencies, the patient's burden will be reduced, and we will really improve our humanity, in that sense. Thank you.
Yeah, just one line up, Manoj. So AI, as I said, right, could help. And then I think India could be in advantageous position, because we are powerhouse of the whole software industry, and then also they are developing a lot of AI companies in India, right? As we are seeing that. So I think innovation in this space where India was not very competitive for a long time, this could be our opportunity to get into that.
Thanks, Uday. Thanks, Bhushan. From my perspective, who has worked in AI but not in this area, I would say two messages, right? Drug discovery and development is not just a business; it's a personal touch, touching every person else there. So anything that we do can help people's life, live healthier life, get cure is that thing. Second part is I think agentic AI is going to help, but it's going to be a co-pilot. The need for scientists, the need for chemistry knowledge, the need for biology knowledge, the need for data science knowledge is going to increase. It may not be same level. You may not have the basic level need, but it's going to increase. So anybody who is looking into any of that part, I think they should be excited. They should get trained. They should look at the latest model. They should look at what's happening, and they should ramp up their skills because a lot of exciting things are coming, and it's just going to be a amazing growth in AI and drug discovery going forward. So I want to thank Uday, Bhushan for fantastic part. Thanks a lot. Uh, One last thing before we kind of say bye is hopefully you guys enjoyed this channel. Hopefully you enjoyed the webinar. As Madhavji said in the start, Garje Marathi is a community of professionals, entrepreneurs, students all across the world. So we hope that you like the webinar and we hope you become member of Garje Marathi. The membership is free, and once you become the member of community, you get a lot of exciting news. You get a lot of insight discussions, chatters. There are internal groups that are part of that. So please take a opportunity to join Garje Marathi as a community. Again, the membership is free, and it's an amazing community, worldwide community, and please take a time. So with that, I do want to thank all the people who made this panel possible. Madhavji, this is your vision about GMG Select Talk. Thank you for taking this initiative to come with a wonderful. Prasadji, your support is very silent but very crucial. Without you, we cannot do that. I know there are other people in Pune office and other areas who work silently and who make this. These are the foundation that allows us to continue this part. So thank you everybody from our heartful. And last but not least, thank you all the audience. Hopefully you got something out of it. We hope you engage with us one way or the other on LinkedIn, on Garje Marathi channel or other way. Thank you. Madhavji, please take it.
Thank you very much. I just wanted to express my sincere thank to you, Manojji, because you have been thanking everyone else, but your efforts cannot go thankless. So thank you very, very much. I really appreciate you taking time and coordinating. It is not easy to coordinate with the panel, and I'm sure that it takes a lot of efforts and it takes a lot of perseverance to follow up and to get right things out of them from the webinar. So I really appreciate each one of you. Thank you and good night. Bye-bye.
Thank you.
Bye.
Bye-bye. Bye-bye.
Bye.